All right. Good afternoon. My name is Jordan Rattan, and today I will be giving a presentation on the processes of glycolysis and gluconeogenesis. Here are our objectives for these two processes. First, we're going to talk about glycolysis, kind of what it is, what makes it up. We'll go over like the steps of glycolysis in pretty good detail. Phase 1 and phase 2, we'll break it down a little bit, and we'll go over like the summary and kind of the net result of glycolysis. Okay, that's what we'll do first. And then we'll go on to looking at gluconeogenesis. You might notice that the process of gluconeogenesis, I don't have a lot of bullet points underneath it. And so as we're going through this presentation, you're going to see how glycolysis and gluconeogenesis are connected to one another. And by understanding one process, you end up with a very decent grasp of how the other process works. So we'll talk about both of them, but we'll go into glycolysis. We'll go into a lot of detail with it, so that way you can understand gluconeogenesis. And so by the time we get to the end, we'll do like a very short, quick comparison of the two processes and kind of what makes them up. Okay, how do they work and how do they tie together? So here's a quote from Regulation of Glycolysis and Gluconeogenesis. Recall that glycolysis and gluconeogenesis are both pathways of glucose metabolism that produce opposite results. Glycolysis is the breakdown of a molecule of glucose into two molecules of pyruvate and free energy. Gluconeogenesis, on the other hand, is the generation of glucose from two molecules of pyruvate and other substrates. In the body, these two pathways are reciprocally regulated, meaning that situations that activate one pathway also can inhibit the other pathway. Okay, so just from this quote, you probably have a good idea of what's going on already, right? So we're looking at how glucose is being broken down to form energy, and in the process, it makes pyruvate. Okay, that's the process of glycolysis. When we start looking at gluconeogenesis, it's kind of like the opposite process, right? Like we're wanting to make glucose from pyruvate. And so it's the opposite of what glycolysis is trying to do. Um, and like it says, it's reciprocally regulated. They go kind of hand in hand with one another. There's a lot of similarities between the two processes. However, um, they're going to inhibit one another whenever one pathway is activated, right? Like both things can't be happening in a cell at the same time. So they're going to work together. They're going to work against each other. But it all comes together to make energy in our body, and that's kind of the overall goal. Okay, so let's start looking at glycolysis. So if you look over onto the right, you can see already that this is a pretty, elaborate process right here. It's got a lot of steps. There's a lot of chemistry happening, and so I'm going to do my best to try and explain all of the chemical processes that are occurring in glycolysis. But this is a summary of the overall process, and don't let this frighten you too much. We're going to be breaking it down quite a bit to make sure we all have a good understanding of what's going on. So first thing you need to know about glycolysis is it is a process that uses enzymes to break glucose down into pyruvic acid. Okay, and this is a process that we find in most living organisms, and it occurs within the cytosol of our cytoplasm. Okay, within our cells, and these are usually when we're looking at these cells, we're looking at our muscle cells, right? Because our muscles are really what need a lot of energy in our body. And so our muscles are constantly taking in glucose and breaking it down. That way they can get the energy release that it needs in order to keep pumping, right? And of course, obviously, the more movement we're doing, the more exercise we're doing, physical labor, whatever, the more glucose needs to be broken down. And so that's kind of the process we're looking at. And this process occurs within the cytoplasm because you think about cytoplasm, right? Cytoplasm kind of contains a lot of ingredients. Well, it does contain a lot of ingredients that the cell needs, right? Like it's going to have a bunch of enzymes, sugars, that glucose we're looking for, right? All these important chemicals are found in very high concentrations in the cytoplasm, right? Because that's how it's able to enter and exit the cell. And so this is where the breakdown of glucose will occur. So, Whenever we're looking at glycolysis, we have to remember, yes, this is breaking down glucose, it's releasing energy, but it's also the first step of cellular respiration. And so it plays a big part in that. So this really kicks off that process, which we know goes on to the citric acid cycle and then oxidative phosphorylation. Okay, but this is the first step for cellular respiration. Um, and like I said, it's used to extract energy from glucose. And so when we start looking at glucose, we're breaking it down. We're going to split it, kind of our main goal. We're going to split it into two 3-carbon molecules, okay? Two molecules that have 3 carbons in them. That's going to be our pyruvates. And our pyruvates are just the simplest structure out of all the alpha keto acids that we find in our bodies. And we know that pyruvates are involved in several metabolic processes within the cell, not just glycolysis, but it is very important for glycolysis as well. Glycolysis is an anaerobic process. That means it does not require oxygen. You think about what you know about the anaerobic processes already that occur. Whenever oxygen is not involved, right, in a process, we usually end up with a high energy product, right? And so by glycolysis, not using oxygen, but we're breaking down this glucose, we're getting a lot of energy that's going to be produced by the end. We want to, right, we don't want a process that's going to give us a very limited amount of energy, right? Because then we would be burning glucose all day long. We want something that's going to contribute more energy than it's being used up for this process. So every reaction that's going to occur um, in glycolysis is going to be catalyzed by an enzyme. Okay, so keep an eye out for those enzymes, especially phosphofructokinase. This is the most important enzyme that we're going to talk about in glycolysis. That's because it's catalyzing the formation of fructose 1,6-biphosphate. If you know what that is, that's the Hardin-Young ester. Just going to tag that in right there. But this is the fructose 1,6-biphosphate. This is what most of our glucose and fructose, whenever it passes into the cell, it's pretty quickly going to be transformed or transcribed into this other type of sugar because it's much easier for our body to break down. And as we're going through this process, you'll see where it kind of comes into play. If you're looking ahead, you can kind of see it's popping up over here after step number 3. That's where our energy is really kickstarting the process. This is where our energy, a lot of it's going to come from. And it's important for this process because this is what's going to allow us to our cells to speed up or slow down the process of glycolysis depending on how much energy our cells need. So if our cells, if we're working out, we're working those muscles, our cells need a lot of energy. It's going to start converting the glucose and the fructose that's passing into the cytoplasm. It's going to start converting it pretty quickly into that fructose 1,6-biphosphate. But if we're resting, it's not going to be so readily converting it over, right? Our cells are going to be more relaxed, just depending on those energy needs. Okay, so let's start talking about our 2 phases. So we know our substrate is D-glucose, right? That's what we're going to start with. It's going to kick off our process. And we're going to go into 2 different phases, and we're going to call them energy investment and energy generation, or phase 1, phase 2, whichever is going to be easier for you to remember. So the same picture right here, I've broken it down into its 2 different parts, okay? We're going to look at phase 1, otherwise known as energy investment, and kind of how we're having to put energy into the cell in order to generate more energy. And then we'll look at the energy generation part, okay? So we're going to look at the first 5 steps of glycolysis. There are 10. Okay? So energy investment. Our glucose is going to become rearranged. Okay, that's the first kind of big part. We got to rearrange glucose, and we got to rearrange the 2 phosphate groups that are attached to it. Those phosphate groups carry a ton of energy in their bonds, and we're going to see how they kind of affect the energy production. But as we start rearranging glucose, it's going to create that fructose 1,6-biphosphate. This is the modified sugar. And if you need to, and this right here, this is all I'm kind of telling you. I'm giving you a quick overview before we start breaking it down, okay? So we're taking glucose, we're going to start breaking it down. We eventually want to get to this fructose 1,6-diphosphate, right? That's what fructose and glucose are kind of readily converted into as they enter the cell, because that's what's going to control how much energy the cell needs. It's going to speed up or slow down the process. And so as we start modifying glucose into this modified sugar, eventually this modified sugar is going to split into two, okay? We get two 3-carbon sugars, but they're different from one another. So we're going to get glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, okay? This is your overview, okay? This is where we're getting to. This is just the energy investment phase. So now let's break it down. Let's go over it little bit by little bit, okay? So over here, we've got glucose. Okay, this is what we're starting at. This is a C6H12O6 molecule. Okay, so we're starting with glucose. We need to convert glucose into glucose 6-phosphate. That's going to occur because of the enzyme hexokinase. Okay, so hexokinase comes into play. It's going to use ATP to get this process started. But as you remember, we're trying to create energy, right? But we've got to invest some energy before the process can get going. So we're going to say we kind of have negative energy right here, okay? So as we invest a molecule of ATP, right, it's going to be converted into ADP, and that's what's going to convert glucose into glucose 6-phosphate. So already in our trek to produce more energy, we've already used up one molecule of ATP. Okay, so we've got some energy debt here already. Okay, so we've used up a molecule of ATP, but we do have glucose 6-phosphate. Okay, and if you notice, it's not terribly different over here from our glucose, but we've got a phosphate group added on right there. Okay, remember, a lot of energy is carried in these phosphate groups. Step 2, we have phosphoglucose isomerase or phosphoglucomutase, and that's another enzyme that's going to come into play that's going to cause our glucose 6-phosphate to rearrange into fructose 6-phosphate. Okay, aldose is converted into a ketose. We've got our phosphate group still sitting down here, but you might notice we've got a little bit of a change up here. So now we've got fructose instead of glucose. Okay, we've got phosphofructokinase, and it's going to come in, it's going to phosphorylate our fructose 6-phosphate, and it's going to cause it to form fructose 1,6-diphosphate over here, okay, or biphosphate. And so as we're adding in energy to continue this to happen, remember I told you to keep an eye on this phosphofructokinase. Here we're adding in a second molecule of ATP. It's getting converted into ADP, so now we've got an energy debt of 2. Okay, we haven't even made any ATP yet, but we have an energy debt of 2. We've already used up 2 molecules of energy. But now we're down to this fructose 1,6-diphosphate. This is what we really want to be at, right? This is what's controlling how fast this process is occurring, depending on how much of this we find in the cell. So glucose, fructose is readily getting converted over into this, so that way this process can begin. But now that we're into this fructose 1,6-diphosphate, it's going to split as well, okay? Whenever it splits, it's going to form 2 3-carbon molecules that are different from one another, and I already mentioned them down here, and you can see kind of their structure. Okay, we've got glyceraldehyde 3-phosphate, dihydroxyacetone phosphate. Okay, so the dihydroxyacetone phosphate, it cannot enter into the next reaction. Okay, so whenever we start looking at phase 2 kickstarts with glyceraldehyde 3-phosphate. Okay, step 4 is fructose 1,6-diphosphate breaking down. This is what's going to kickstart that next reaction. This is the substrate we're going to use. Okay, so when we have dihydroxyacetone phosphate, you're sitting here like, oh, what are we supposed to do with that? Like, where's that going to go? Well, usually what the cell's going to do is going to use the sorry, it's a big word, triocephosphate isomerase. Okay, this word down here, big word. And it's going to use that to convert the hydroxyacetone into the glyceraldehyde 3-phosphate. Okay, we got to convert this so that way it's not just piling up in the cell, because otherwise we'll have a lot of just piling up. It would cause bad effects. We don't want that. It's got to be converted over into the glyceraldehyde. It gets converted, it can go on to the next process as well. Okay, this is just kind of setting up for our big energy production. Remember, we do have -2 energy right now. We've used up 2 molecules of ATP, yet we haven't created any yet. So in glycolysis phase 2, otherwise known as the energy generation phase, you might notice this is that same picture, just kind of cut up a little bit. I went ahead and threw on step 4 and step 5. Step 5 is just converting this dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. That's all step 5 is. Once we get enough glyceraldehyde 3-phosphate, the process can begin. Okay, so once again, I've got kind of the general outline down here of this process, if that's easier for you to look at and understand, but there's also the images up here if you need to understand kind of how the structure changes by looking at it. And so all of this glyceraldehyde 3-phosphate, okay, step 6. So this is going to be oxidized by glyceraldehyde 3-phosphate to form carboxylic acid. Okay, so this is going to produce our 1,3-biphosphoglycerate. Okay, 1,3-biphosphoglycerate. So this is oxidizing. We've got NAD positive is going to be reduced to NADH. The phosphate group comes over, it attaches through a very high energy bond right here. Like I said, keep up with this phosphate. Okay, that's going to produce a lot of energy here in a little bit. So this is step 6. Okay, we're converting it over into this 1,3-biphosphoglycerate. Okay, NAD has converted into NADH. Step 7, we're harvesting ATP finally. Okay, remember right now we're at -2 ATP. We've used up 2 molecules of energy. We need to start getting some back. So we're going to harvest some ATP. Okay, if you notice here, ADP is being converted into ATP via phosphoglycerate kinase. Okay, so this is also going to transfer our very high energy phosphate group over here to ADP. We produce 3-phosphoglycerate, and this produces ATP in the process. Okay, so keep up with our phosphate groups. There's one up here, we've got one over here. Because we've got rid of this phosphate group, energy's been produced. Okay, you see how that works? As we get rid of this and we break this bond, energy is getting produced. We're getting ATP. And that's kind of our ultimate goal. And so this whole process right here, step 7, going from 1,3-biphosphoglycerate to 3-phosphoglycerate, we're losing a phosphate molecule. And this is the very first substrate level phosphorylation that we see in glycolysis. Okay, this is the first time we're getting energy back after we've already invested 2 ATP. We're finally getting one back. So now we're just in the -1 ATP. So then we come on, so we got the enzyme phosphoglycerate mutase, and it's going to isomerize 3-phosphoglycerate, and it's going to form 2-phosphoglycerate. Keep it on this phosphate group. It's traveling from our carbon 3 to carbon 2. Okay, so it goes from third carbon, which you see up here, it's transferred up here to the second carbon. Okay, remember, that's what's going to create more energy here in a second, so keep an eye on it. As we're going into step number 9, the enzyme enolase is going to act as a catalyst, and it's going to actually dehydrate. So when we say dehydrate, we're creating a molecule of water, and it's going to travel into the cell. So we're creating a molecule of water. We're dehydrating our phosphoglycerate. So now we're going to have phosphoenol pyruvate. Okay, so we've gotten rid of a whole water molecule. Okay, if you look over here, you can see there are a couple H's and an O shorter. Okay, because we lost this molecule. And so phosphoenol pyruvate is very energy rich. This is the highest energy phosphorylated compound found in metabolism. And so as we're coming over this direction, what do you notice? What have I been saying? Keep an eye on this phosphate group. Where'd it go? You don't see it over here, do you? So what's going to happen in step 10 is pyruvate kinase is going to take that phosphoryl group, and it's going to give it to ADP, and it's going to create ATP. Okay, and it's going to also release water in the process. But that's how we're going to get to that pyruvate. Okay, so what have you seen? We lost 2 molecules of ATP in the first phase, but here maybe you're saying, "Hey, Mr. Tan, what's going on? I noticed we've only made what 2 molecules of ATP. We're even, right? We're out of debt. We're even. How I thought we were going to make energy, right? We're using energy. We need to make energy. Okay, well, also you might want to think back to this step right here. So whenever we break down this molecule up here, right, it forms these 2,3-carbon sugars, and they're different from one another. So since we've got 2 sugars here, what do you think is going to happen? Well, this glyceraldehyde 3-phosphate is going to start coming through this process, and it's going to produce what? 2 ATP. Well, we've got dihydroxyacetone phosphate over here. It's going to also get converted into this, and it's got to go through this process as well. So for every 2 glucose, remember up here, it's going through a process. It eventually breaks down into these 2 molecules. We're going to have this one go through, then this one will go through as well. So we're actually going to create 4 molecules of ATP. Okay, and that's what our summary of glycolysis is. I want you to see this whole process together. As we're going from glucose, we take a molecule of ATP, take a molecule of ATP, this fructose 1,6-diphosphate breaks down into our 2 different sugar molecules down here, and they both need to go through this process. As they go through, we're making ATP, So that's 2 ATP, right, for the first sugar. Second sugar goes through, we make 2 more, okay? So we end up doubling, when we take 2, we end up with 4. We're going to double the amount of energy that we took and that we used up. Okay, so this is just the net result of glycolysis. This is kind of the, if you need to see it in this kind of format, if that helps you, this is the net equation up here. Well, here's our net products, right? We're going to have 2 pyruvic acid molecules, those 2 pyruvates we end up with at the end, 2 NAD positive molecules. If you're paying attention to those guys, let's see. Those are going to be converted down here as well. And we're going to have 2 ATP molecules, right? But remember, when I say 2 ATP molecules, that's per already broken down molecule of the fructose 1,6-biphosphate. So we're going to actually have 4 ATP molecules here at the end because of that process. Okay, so we're doubling the amount of ATP we're creating. That's glycolysis. I know that took a while. I really wanted you to understand that process because gluconeogenesis is much easier to understand once you already kind of have a good idea of what glycolysis is. So gluconeogenesis is going to form glucose from non-hexose or non-carbohydrate precursors such as glycerol, lactate, pyruvate, gluconeogenic amino acids, that kind of stuff. So basically, we're kind of doing the opposite of what we just did in glycolysis. We're going to convert pyruvate into glucose. Okay, and so I want you to pay attention to what I'm saying, right? I want you to listen to my words. This is the opposite process of glycolysis, right? Glycolysis goes from glucose to pyruvate. Now we're going from pyruvate to glucose. But this is not a reversal of glycolysis. It's an opposite process, not a reversal. Okay, so like I said earlier, if you understand glycolysis, gluconeogenesis is easier to understand because there are many processes, many steps that are very similar whenever we start looking at how we go from pyruvate to glucose. However, there are some exceptions. There are going to be a few steps that are a little bit different. And so that's kind of what I want us to focus on because if we already understand glycolysis, we can understand gluconeogenesis. We just need to know where the differences are. Okay, so let's start looking at that. So as you start looking at this, okay, we're going from glucose up here down to pyruvate. You see some of the same words that you saw a minute ago, it's just set up a little differently. Okay, so these steps, they're similar, right? And we're not going to go over all of them again because we just did that with glycolysis, but we need to start looking at the exceptions. So whenever you see like this red, this is glycolysis enzymes. But whenever you see blue, that's gluconeogenesis enzymes. Okay, that's where the differences are going to come into play. Okay, so I want us to mainly look at the non-reversible steps of glycolysis. And luckily for us, there are only 3 non-reversible steps. Okay, so everything else will be reversible except for step 10, step 3, and step 1 mentioned earlier. Okay. And so let's go ahead and like break those down a little. So you might say, why is step 10, step 3, step 1, why are they out of order? Well, remember, we're going backwards now. Now we're looking at gluconeogenesis. We're reversing this process. So I want us to look at this in the reverse process as well. Okay. So since these processes cannot be just reversed, they have to be bypassed with new routes. And so the first step, step 10, okay, this is going to be step 1 for gluconeogenesis. Pyruvate cannot just be converted into phosphoenol pyruvate. Okay, a different process has to occur. So there's going to be 2 enzymes involved at this point, and they're going to catalyze this reaction, right, using gluconeogenesis. And so these 2 enzymes are pyruvate carboxylase and PEP carboxykinase. And so they're going to come into play, and instead of pyruvate just becoming phosphoenol pyruvate. As you can see, pyruvate has to be converted into oxaloacetate. Okay, we use a molecule of ATP here. You notice that using a molecule of ATP, and then it's also going to be converted over here into the phosphoenol pyruvate. Okay, so we can't just go reversal, right? We've got to add in a molecule of energy to form oxaloacetate to form that phosphoenol pyruvate. And then the process can continue, right? Well, that's what we're trying to get to, isn't it? That step that we're trying to get to this, we're trying to, we're trying to get the pyruvate to our glucose. There we go. Sorry, I was mixing it up in my head a little bit. It's a lot of chemistry, guys. And so this is our first step. Okay, we get it converted and we can go through step 2, easy peasy, not a problem. We get to step 3, fructose 1,6-biphosphate converting to fructose 6-phosphate. That's not a process that's just reversible, okay, in glycolysis. It's got to go through a couple of steps. And so this is actually going to occur via hydrolysis, okay, to reverse it, right, for gluconeogenesis. So fructose 1,6-biphosphate is going to become stimulated by the high amount of ATP. The higher our ATP in the cell is going to favor gluconeogenesis. And so our fructose 1,6-biphosphate, in order to go back this direction, what do you notice? What's changed? This phosphate group right here. Okay, so since we've broken down glucose, right, through glycolysis, we're going to have a high amount of ATP at the end. Since there's a high amount of ATP, maybe we want to convert some of that pyruvate back into glucose in order to conserve ATP. So we reverse the process. So all this ATP, right, we're going to take this fructose 1,6-biphosphate, watch that phosphate group, where's it go, and convert it into fructose 6-phosphate. Okay, we still have 1 molecule of phosphate. That one's gone, but now we've got a molecule of ATP. Okay, and then we can keep going steps all the way down to step number 1 or step 10 in gluconeogenesis. The very last step for gluconeogenesis. Glucose 6-phosphate can't just readily be reversed into glucose. Okay, it's going to take, it's going to also occur via hydrolysis. Remember, when hydrolysis occurs, we're producing a molecule of water, H2O. So it's going to occur via hydrolysis as well. We create a molecule of water, and this becomes catalyzed by glucose 6-phosphate, and it's going to come in, it's going to be converted into glucose with a PI left over. Okay, so these are our 3 non-reversible steps of glycolysis for gluconeogenesis. And this is just a diagram to kind of help you visualize, right? So glycolysis, we're breaking down glucose all the way down to pyruvate. But if we want if we want to see the opposite, right? Gluconeogenesis is pyruvate going back to glucose. This is showing you the irreversible steps where things are having to change. So we can't just readily convert back. We've got to use this enzyme. You got to use this. right? We have to add in these extra things, this extra energy or whatever, to reverse this process. Well, not reverse, but to have the opposite process occur. Okay. And this, once again, is kind of showing you glycolysis is happening this direction, gluconeogenesis is happening this direction. The blue is going to be the enzymes used during glycolysis. The red is the enzymes used during gluconeogenesis. These are opposite processes from one another, not reversal. We have to have extra enzymes and extra steps um, in order to have pyruvate converted back into glycolysis. That's why we say it's not a complete reversal, right? It's just the opposite process. It's going to take a little extra. It's its own process that occurs, but they do share many parts, many similarities, and that's why we kind of lump them together when we're trying to explain these processes. All right, well, I hope this helped you out a little bit. I hope you were able to understand these two processes a little bit better. They're very similar. So thank you for taking the time to listen to me today, and I hope you have a very blessed day.